Hello everyone, and thank you for joining our webinar, Spatially Resolving the Unknown with Unbiased Whole-Transcriptome Mapping. My name is Renee, and I am an application scientist in the Academic Affairs and Innovation team at M20 Genomics. Today I'm excited to introduce our new spatial transcriptome platform and share how unbiased whole transcriptome profiling can help us explore biology beyond the limitations of conventional spatial approaches. Over the next 30 minutes or so, we'll take a look at the technology and explore some of its applications, from archived FFP samples to complex host pathogen interactions. So with that, let's get started. Spatial transcriptomics has transformed how we study gene expression within intact tissue. But to fully understand biology in its spatial context, measuring messenger RNA or mRNA alone isn't enough anymore. Tissues are made up of diverse cell states, organized into complex spatial niches. And a lot of what drives biological processes goes well beyond protein-coding transcripts. Non-coding RNAs, microbial RNAs, and viral transcripts all play a real role in disease biology. But with conventional spatial approaches, they're largely invisible to us. At the same time, some of the most valuable clinical specimens we have are preserved as FFPE tissue. But RNA fragmentation in these samples creates a major challenge for conventional assays, which means researchers often cannot fully leverage these rich clinical resources. So all of this just really points to one conclusion, which is that we need a more comprehensive approach to spatial transcriptomics. So what we can discover with spatial transcriptomics is ultimately influenced by the capture chemistry. Many conventional spatial platforms rely on poly-A-dependent 3' capture. This works well for intact eukaryotic mRNAs, but it also comes with some limitations. For example, it's not well suited for capturing bacterial transcripts. It mainly provides information from the 3' end rather than across the full transcript body. It also has limited ability to detect many non-coding RNAs, and its performance can be affected when working with fragmented RNA, such as that in FFP tissue. So instead of relying on intact poly-A tails, we use optimized random primers for total RNA capture. This means that we can capture RNA fragments across the transcript rather than focusing on only one end. And this provides more complete gene-to-body coverage and enables detection of coding RNAs and non-coding RNAs within the same experiment. It's also worth mentioning that some spatial platforms have already addressed the FFP challenge by using targeted probe panels. And although these approaches can improve detection of selected transcripts, because they rely on predefined targets, this means that they're also limited in terms of discovery. With random priming, since we're taking on a different approach, we not only improve detection in challenging samples, but we also expand the range of biological questions that we can address. With a new capture chemistry, validation is essential. Our random priming chemistry has already been extensively validated in our single-cell transcriptomics platform called NATA. We have applied this chemistry across a wide range of applications, including frozen tissue, FFP tissue, cultured bacteria, and complex microbiome samples. These results are supported by both internal validation and multiple peer-reviewed publications. Across these applications, we consistently observe reliable capture of diverse RNA classes across different organisms. This provides a strong foundation for extending the same validated chemistry into spatial transcriptomics. And this is how we developed the NATA Spatial platform. NATA enables unbiased spatial total RNA profiling, moving beyond 3' biased capture and predefined probe-based approaches. NATA allows detection of both coding and non-coding RNAs within their spatial context. The platform is optimized for FFP specimens, enabling sensitive and reliable transcript detection even in challenging archival clinical samples. NATA also supports host pathogen co-mapping within a single tissue section. So this allows us to localize host transcripts alongside bacterial or viral transcripts while preserving their spatial context. NATA provides flexible spatial resolution options, with both 15 micron and 50 micron spot sizes. This allows us to choose a resolution that best fits different biological questions, from broader tissue level organization all the way to higher resolution spatial profiling. So in summary, these capabilities enable comprehensive, unbiased spatial transcriptomic profiling across a broad range of biological and clinical applications. So how does NATA work in the lab? Let's take a look at the workflow. This workflow is designed as an end-to-end process, with spatial information preserved throughout each major step. We first start with a prepared tissue section, for example, a 5-micron FFPE section, and it's mounted on a standard adhesive glass slide. We deparaffinize and then permealize the sample, which allows the molecular access while maintaining the tissue architecture. The next step is random primer-based in situ reverse transcription. This is an important part of the workflow because the first-strand cDNA is generated directly within the tissue section. This allows the spatial location of the transcripts to be preserved from the beginning of the process. The synthesized cDNA is then transferred and captured on the spatial capture chip. To do this, we need to use the NATA Align spatial instrument. This provides precise spatial alignment during the cDNA transfer while maintaining the original tissue coordinates. Following the capture, the cDNA then undergoes extension and elution, followed by library construction and then next-generation sequencing. These core workflow steps are integrated into the NATA Omni reagent kit, which gives a standardized workflow from tissue input all the way through library preparation. Now let's move on to some data. Here is data from a mouse brain FFPE sample. As I mentioned previously, FFPE is one of the more challenging sample types for spatial transcriptomics due to the RNA fragmentation. And even in this challenging setting, at the standard 50-micron resolution, NATA achieved a median of over 46,000 UMIs and nearly 6,000 genes per spot. We can also clearly resolve the expected anatomical structures across the tissue. We detected nearly 34,000 genes in this single mouse sample since NATA captures the whole transcriptome rather than only protein-coding transcripts. This gives us a broad range of information for downstream analysis. Here we're looking at a human oral squamous cell carcinoma FFP sample profiled at 15-micron resolution, which is at a higher spatial resolution and much smaller spot size. We achieved a median of more than 10,000 UMIs and approximately 3,700 genes per spot. This shows near cellular spatial resolution while maintaining strong transcript detection sensitivity. It's important to note that this was a high-quality sample sequenced at a much higher depth than our standard workflow. So these values represent an upper range of our performance scenario. Under standard sequencing depth, we typically observe more than 1,000 median genes per spot at 15-micron resolution, which is still very strong transcript detection. Across the entire tissue section, we detected more than 57,000 genes in total. So together, these two datasets demonstrate that NATA provides robust spatial transcriptome profiling at both 50 and 15-micron resolution, while still maintaining strong sensitivity in FFPE samples. Although UMI and gene counts provide important measures of performance, they don't show the full picture. So we also examined saturation curves across different spatial resolutions. On the left, we show a human breast cancer FFP sample profiled at 50-micron resolution. On the right is the oral squamous cell carcinoma FFP sample that we saw previously at 15-micron resolution. In both datasets, the median number of detected genes per spot continues to increase as more mapped reads are added, without reaching an early plateau. So this tells us that deeper sequencing continues to recover additional transcript information rather than primarily increasing duplicate reads. Both samples show consistent saturation behavior despite differences in tissue type and spatial resolution, which demonstrates our ability to generate high complexity libraries across different tissue types and spatial resolutions. To evaluate NATA's performance in clinically relevant samples, we applied the platform to multiple archival FFP tumor specimens. Here we show representative spatial clustering results from human breast cancer, ovarian cancer, and endometrial cancer samples. Across these different tumor types, NATA generates high-quality spatial transcriptomic profiles and enables the identification of spatially resolved molecular features within diverse clinical FFP tissues. So these results show the feasibility of applying NATA for spatial profiling of clinically relevant FFP tumor specimens. We then evaluated NATA's performance across a broader range of tissue types, summarizing key performance metrics across multiple FFP samples and spatial resolutions. So at 15 micron resolution across both mouse and human tissues, including brain, embryo, breast cancer, ovarian cancer, and endometrial cancer, we consistently achieved median gene detection in the range of around 5,000 to 8,000 genes per spot. This consistency was observed across diverse tissue types with different biological characteristics. At higher resolution, using the 15 micron workflow, we also maintained strong transcript detection. Both mouse olfactory bulb and human oral squamous cell carcinoma samples achieved more than 1,000 median genes detected per spot. We then evaluated spatial fidelity by comparing NATA-derived spatial expression patterns with established anatomical references. In the mouse brain at 50 micron resolution, we looked at canonical neuronal markers, including GAD2, SLC17A6, and synaptotagmin1. Their spatial distributions closely matched the expected organization of excitatory and inhibitory neuronal circuits. When we compared this with in situ hybridization data from the Allen Mouse Brain Atlas, NATA showed strong concordance with established anatomical patterns. We also looked at the mouse olfactory bulb at 15 micron resolution. The neuronal and synaptic markers recapitulated the expected layer-specific organization associated with sensory processing, which again were consistent with in situ hybridization results from the Allen Mouse Brain Atlas. So these results show that NATA preserves biologically meaningful spatial expression patterns while still maintaining spatial fidelity across different resolutions. We next examined how NATA's random priming chemistry impacts transcript coverage along the gene body. So with conventional oligo-DT capture, shown in the green, sequencing reads are strongly enriched toward the 3' end of transcripts, so this results in limited coverage across the rest of the gene body. On the other hand, NATA's random priming chemistry, shown in the orange, produces a much more uniform distribution across the transcript, with a much more reduced 3' bias and a more complete gene-to-body coverage. This broader coverage preserves the transcript-level information beyond simple 3' counting and enables additional analyses, such as gene fusion detection and alternative splicing analysis. So NATA generates spatial transcriptomic data that supports more comprehensive molecular characterization. Building on the expanded gene-to-body coverage enabled by random priming, NATA extends its range beyond protein-coding transcripts to capture a broader range of RNA species. Over the past decade, non-coding RNAs have become increasingly recognized as important regulators of gene expression and cellular states. They have been implicated in processes including development, disease biology, and emerging therapeutic strategies. But understanding their spatial distribution within tissues still remains challenging. Here we quantified the diversity of RNA species detected in a mouse brain section. On the left-hand side, NATA detects not only protein-coding RNAs, but also a large range of non-coding RNA species, including link RNAs, microRNAs, and other regulatory RNA-associated species. On the right-hand side, we examined the spatial distribution of these RNA species within the same tissue section. These non-coding RNAs show distinct spatial patterns rather than random distribution, so they suggest their organization within native tissue architecture. This expands spatial transcriptomics beyond protein-coding gene expression and provides new opportunities to investigate spatial regulatory mechanisms involved in disease biology. We next applied NATA to a clinically derived FFPE oral squamous cell carcinoma sample to investigate spatial tumor state organization. On the left is a histological image, where a specific region has been outlined based on morphological features. And on the right-hand side, we mapped key markers and functional programs, including MKI67, TP53, Cestrin-1, as well as cell cycle, EMT, and quiescence signatures. The spatial distribution of these tumor state programs closely aligns with the histological features. And within the outline region, TP53 and Cestrin-1 expression are elevated, which is consistent with a stress-associated tumor niche. At the same time, cell cycle activity is reduced, and EMT program activity is lower, and the quiescence signature is relatively increased, indicating a low proliferative tumor state. Interestingly, within this broader low proliferative region, we also identified 2 localized MKI67 hotspots. These correspond to a focal activation of the cell cycle program, showing spatially restricted proliferative subregions within an otherwise non-proliferative tumor area. Therefore, NATA enables resolution of spatial tumor state architecture in FFPE clinical samples, capturing both dominant regional states and localized functional heterogeneity. For this next case, we applied NATA to matched FFPE breast cancer and adjacent tissue from the same patient to evaluate whether established clinical and molecular features are preserved in spatial transcriptomic profiles. For each marker shown here, the top represents the tumor tissue and the bottom represents the matched adjacent margin. The tumor shows strong ERBB2 expression together with reduced ESR1 and progesterone receptor expression compared with the matched margin. So this is consistent with the expected characteristics of HER2-positive breast cancer, which shows that NATA can recapitulate clinically relevant subtype features spatially. NATA also resolves spatial organization within the tumor microenvironment. IgHG1 and CD163, which are immune-associated markers, show distinct spatial expression patterns, which reveal heterogeneity in the immune landscape within the tumor. Interestingly, while IgHG1 is broadly expressed across the tumor region, we identified a localized area with reduced IgHG1 expression that spatially overlaps with the MUC-LC enrichment. This suggests localized epithelial immune heterogeneity within the tumor microenvironment. We also performed spatial deconvolution using matched Vita FFPE single-cell transcriptomic data as a reference. On the left is the tumor, and on the right is the matched adjacent margin, and we're looking at the underlying cellular architecture. As you would expect, the tumor is enriched for epithelial cells, while the adjacent margin has a much larger fibroblast compartment. Within the tumor itself, epithelial and fibroblast populations actually occupy pretty distinct spatial regions. So we're really looking at the structural organization of the tumor microenvironment layout. Then the deconvolution results also support the gene expression features we saw on the previous slide. Plasma cells are markedly enriched in the tumor, and that matches the broad IgHG1 expression pattern we just talked about. Macrophage distributions track with the CD163 signals we saw in both tissues. On top of that, the tumor shows increased T and NK cell abundance compared to the margin, so we're looking at distinct immune landscapes between the two. So NATA isn't just capturing spatial gene expression, it's also resolving the underlying cellular composition. And we get this very coherent view from molecular markers all the way to tissue architecture within FFPE specimens. Another key capability of NATA is spatial host pathogen co-mapping. We analyzed a mouse lung infected with Klebsiella pneumoniae. The left-hand side compares an uninfected control with infected tissue. And with NATA, we were able to both map bacterial RNA abundance along with host immune responses, including neutrophil recruitment inferred through spatial deconvolution. In the infected tissue, bacterial RNA is concentrated in discrete spatial hotspots, which overlap with regions of neutrophil infiltration. We quantified this relationship in the upper right panel over here and observed a strong spatial correlation between bacterial burden and neutrophil recruitment. The host immune response also shows spatial enrichment that corresponds with these bacterial hotspots, which indicates localized immune activation at sites of infection. This demonstrates the ability of NATA to capture pathogen localization along with corresponding host response within a single spatial assay, which provides a direct approach for studying host-pathogen interactions in their native tissue context. This next case examines a hepatitis B or HBV-associated hepatocellular carcinoma. Here we integrated viral infection, tumor evolution, and host immune response within a single FFPE tissue section. We first mapped the spatial distribution of HBV RNA. In the upper left panel, viral transcripts are strongly enriched within the tumor region and largely absent from the adjacent non-tumor tissue, indicating localized viral activity within the tumor microenvironment. We then asked whether this viral distribution was associated with specific tumor populations. We were able to infer spatial copy number variation, or CNV, and identify distinct tumor subclones with NATA's full gene-to-body coverage. The blue arrows in the upper right panel indicate the spatial region occupied by subclone 2. Interestingly, HBV RNA is not uniformly distributed across malignant cells. It actually preferentially localizes to this specific CNV-defined subclone. This enrichment is quantified in the lower left panel, where the blue dashed box highlights subclone 2. We also examined the associated host immune response. In the lower right panel, the complement-associated immune program shows spatial enrichment in regions corresponding to the HBV-associated subclone 2. This spatial co-localization suggests a potential relationship between viral activity, local immune signaling, and tumor evolution within specific malignant subpopulations. So our platform is able to integrate viral infection, clonal architecture, and immune response analysis from a single FFP tissue section, which would be challenging to reconstruct using separate workflows. So we looked at the analytical capabilities of NATA across several biological applications, and they couldn't be achieved without the support by the experimental infrastructure. The first component is the NATA thermal control system, which provides precise multi-step temperature control during spatial transcriptome reactions. This ensures that each reaction condition is tightly controlled and reproducible. Our second component is the NATA-aligned spatial instrument, which enables precise physical alignment during the cDNA transfer step. This allows consistent transfer of spatially resolved cDNA while maintaining the original tissue coordinates. So both these hardware components establish a standardized workflow that integrates precise thermal control with spatial alignment and supports consistent performance across samples and experiments. Generating high-quality spatial data is only the first step. The next challenge is actually transforming that data into meaningful results, which is the role of NATA Insight. NATA Insight runs locally and allows data processing to remain within the institutional environment and provides greater control over sensitive datasets. The software also supports H&E-guided ROI annotation, enabling spatial transcriptomic signals to be directly interpreted alongside tissue morphology. This allows researchers to understand not only which genes are detected, but also where those expression patterns occur within the tissue. The automated analysis pipeline takes raw FASTQ files through spatial gene expression matrix generation and visualization-ready outputs. So, how would you start designing a study with NATA? The first step is defining the biological question, because that will guide the overall experimental design. NATA supports a broad range of applications, including FFPE tissue profiling, oncology studies, coding and non-coding RNA analysis, host pathogen interactions, and virus-host RNA mapping. The next consideration is spatial resolution. We offer two resolution options. One is 50 microns and the other one is 15 microns. The 50-micron workflow provides a broader view of tissue organization with higher RNA recovery per spot, while the 15-micron workflow allows higher spatial resolution for investigating finer tissue structures and near-cellular patterns. And if you're planning a pilot study, you should consider several factors like tissue type and fixation history, section quality, desired resolution, sequencing depth, and downstream analysis goals. And based on these parameters, we can then help define an experimental design that suits your research goals. So, in summary, NATA is a spatial transcriptomics platform that is designed to provide a more comprehensive view of biology within tissue context. It takes an unbiased, total RNA profiling approach with spatial resolution across the full transcriptome. The platform is designed to address practical challenges in spatial workflows, including the analysis of archived, clinically annotated FFP samples with strong transcript detection sensitivity. By capturing host, bacterial, and viral RNA within the same spatial assay, NATA enables investigation of complex biological interactions, including infection biology and tumor microenvironment organization. With both 50 micron and 15 micron resolution options, researchers can select the spatial scale based on their biological questions. These NATA spatial products are all available now. Since every research project has different requirements depending on sample type, throughput, and scientific objectives, we welcome you to discuss sample compatibility, resolution considerations, and potential pilot study designs for your specific research needs with us. And with that brings us to the end of today's webinar. Please feel free to scan the QR codes on the screen if you'd like to learn more or connect with us on LinkedIn. Thank you so much for joining us today, and we really appreciate you taking the time to be here. We hope you found this webinar useful, and we look forward to connecting with you.